discussing case studies, and we don't need to be in the lab, and we don't need to be wearing masks when we have to. And so we're going to try this by Zoom. So come to lab during your regularly scheduled time. We'll do breakouts to work through the problems in the case studies. You have a hard copy of the case study, but it's also on Moodle if you want the Word document. And we'll do it that way. Give it a try. I don't see any reason why it wouldn't be just as good as getting together in the lab. Remember that problem set 4 is due this Friday. If you turn it in early, I'm checking. The envelope is now at CSD desk. I will be checking it daily, and if you turn it in early, I will grade it early, and you'll get your results back early. If you turn it in at the deadline, I'll do it on Saturday. Let's see, what else do I want to say about Moodle? So today we're here finishing up on homeostasis and talking more about the control of the kidney and a bunch of clickers and other things for review for the exam that is on Monday. Any questions about what we're doing this week and then Monday next week? Okay. So let's pick up where we left off on Monday. We're going to talk about control of the kidney by 2 hormones, vasopressin and aldosterone. Then I want to cover water homeostasis again because repetition is sometimes helpful, and doing it at the beginning of a class as opposed to the end of the class can be helpful. So we're going to do some review, and then we'll break out into Zooms, and in your Zoom breakouts you can work on the problem set together, or the clickers and other review items that I have in this PowerPoint. My recommendation for working on the problem set together is to focus your energies on working on setting up the problems and not spend a lot of time. So by setting it up, I mean using the right equation, making the transformations of units that you have to use, making sure that you're rounding off to the correct number of significant digits, thinking that way, or not rounding off, but saying how many significant digits should this number have, but not doing the calculations together. Doing calculations is the sort of thing that you can do individually. So my recommendation is to use your time wisely together to be more conceptual, and then get into the calculation details on your own. That's my recommendation. Okay, kidney, nephrons, glomerulus, filtration. We've talked about the things that control filtration. Constriction of the afferent arteriole, constriction of the afferent arteriole, dilation of the afferent arteriole, the macula densa, what we'll talk more about in a few moments. Filtration, generating a fluid that is working through the proximal tubule. The epithelium of the epithelial cells of the proximal tubule work very hard to reabsorb sodium and glucose and some bicarbonate and some other solutes that we're not talking about because I'm trying to be conceptual and I don't want to overload you with details about calcium or phosphate or other solutes. But if you become a nephrologist, which is what a doctor who specializes in kidney disease is called, you learn more about all of these other transport processes that are going on. We're staying focused on sodium, glucose, a little bit of potassium. We'll talk about urea, but we're not talking about a lot of other things. Anyway, reabsorption of sodium and glucose and bicarbonate here in the proximal tubule. 67% of the sodium is reabsorbed here. Water is reabsorbed here because water follows solute. And this is a leaky, water-permeable epithelium. So 2/3 of the water that you need to reabsorb is reabsorbed here. Okay, so now we're moving into the loop of Henle, where the sodium transport mechanisms are designed to build a solute gradient. So out here it's isotonic, 300 milliosmoles per liter, 300 milliosmolar, isotonic with blood plasma. And then as we move deeper and deeper into the medulla, we build a gradient from 300 to somewhere between 600 and 1200 milliosmoles here. And that variance depends on the ability of the ascending limb of the loop of Henle to pump sodium from lumen into the interstitium. So here we're going to get into aldosterone. When we get to aldosterone, what aldosterone is going to do is going to increase the expression of the pumps, the sodium-potassium ATPase and the sodium-potassium 2-chloride cotransporter. And as I say those words, you should be imagining, okay, yeah, that's right. The sodium-potassium ATPase is in that basolateral membrane, and the sodium-potassium 2-chloride cotransporter is in the apical membrane, and together they move sodium and other solutes from the tubular fluid into the interstitium. Not just move, they pump it. And more pumping along this long ascending limb leads to more of a building of a gradient. And less pumping, less aldosterone, less of a gradient. Now, let me just clarify what I just said. Aldosterone is not the pump. Aldosterone is the hormone that changes the expression of the pumps that I just mentioned. Yes?
Is it primarily affecting the sodium, or does it affect other solids?
You know, I don't really know. I'd have to go back and see if aldosterone affects calcium transport or other solute, but I know it affects sodium transport, and that's what we're going to focus on. Yes, so.
I understand like why there's a gradient between like the ascending and descending loop of Henle.
The descending and the ascending, okay.
Why is there like a gradient like deep down? Like what is it just space wise that the gradient is like the concentration is higher at the bottom? Like what creates the
It is a
top
key
down?
concept that the concentration here is higher, the concentration in the interstitium here is higher than the concentration in the interstitium here. That is a key concept. And that happens because of this geometry. And in order to build this gradient, and then in order to be able to use it here, which I'm jumping ahead, we need this geometry. If we just had a linear tube, we couldn't make 1200 milliosmoles. We couldn't do it. So the combination of this loop and the pumps and the equilibrating across one limb of the loop and pumping on the other limb of the loop, that's what creates the high concentration here that we need to use here in order to make a concentrated urine when we are dehydrated.
But is there like a reason why it's more concentrated? Like, I don't know if my question is making sense. Like, why is it more concentrated at the bottom? Just because of the geometry? Like, what is the logic behind that? Like, that's a loop.
So when you say why, are you asking why did we evolve that, or why mechanistically is it higher?
Yeah, like what, is it just because the space is smaller that the concentration will build up more at the bottom than at the top?
Well, okay, let me get to the next slide. So if I could walk through this again, but you could walk through it, and what you can, if you walk through it with pumping and equilibrating and moving some new fluid in and pumping and equilibrating and moving some new fluid in and pumping and equilibrating and moving some new fluid in and pumping and equilibrating. If you do that, all those processes over and over and over again, because it's equilibrating here, it's going to be isotonic here. And because of the pumping and equilibrating, it's going to be hypertonic here. That's just the mechanics of the so-called counter-current multiplier. Counter-current, it's going this way, so this way and this way, right? Going against each other. A physicist could probably explain it from a differential equation perspective, but I can't do that. But the consequence of things going in opposite directions, starting isotonic, pumping and equilibrating, and then moving new fluid in, that combination of things
Okay.
makes it hypertonic down here.
Thank you.
Is that,
Yeah.
now did I answer your question?
Yeah, now,
Yeah. If we didn't have the loop, it wouldn't work, it wouldn't happen. If we didn't have the differential pumping on one side and equilibrating on the other side, it wouldn't happen. The more the pumping, the more it happens. The longer the loop, the more it happens.
My confusion was just like, because it equilibrates, like, wouldn't it be the same, like, all the way down to the fence?
Well, because it's, I guess the short answer is no. It wouldn't be the same because this is the mechanism that works. I mean, it would be the same if it was linear, but it's not the same with counter currents. Does that help?
Thank you.
Other questions? So this, what I want you to take away from this is that the loop of Henle, shallow and deep loop of Henle, use this countercurrent multiplier mechanism, combining pumping. Remember, embedded in this pump is the sodium-potassium ATPase on the basolateral membrane and the sodium-potassium 2-chloride cotransporter on the apical membrane. That combination of transporters represents an ATP-driven pump that pumps, and then the geometry and the flow, that is to say, flow in and equilibrating on one limb, and active transport on the other limb. Doing that over and over again, you build a gradient. And then we're going to use that gradient to make a concentrated hypertonic urine or not, depending on whether we're dehydrated or not. Other questions? This seems like magic, and I know that it's hard. By magic, I mean something that you don't understand. But it's not magic, it's just physics and transport processes. But I'm happy to answer your questions. Okay, well, let's go on and just use the knowledge. So, if you are highly hydrated, you do not want to put out a concentrated urine. You want to put out a dilute urine because you are, in fact, you might need to put out a highly dilute urine in order to get rid of excess water. You might be saying, well, under what conditions would we have excess water? Well, remember, if your body is too hydrated, then your solute concentrations are wrong, they're too low. And low solute concentrations can lead to abnormalities of heart, brain, and other function. So the kidneys have evolved to make sure that we don't do that. So input is 300 milliosmoles. In the setting of high hydration, we have low aldosterone. Low aldosterone means low sodium transport here. So this is sodium pumping. It doesn't say high or low, it happens. But you should annotate this slide by saying low sodium pumping on the ascending limb. And so the counter current multiplier is not optimized. And so we don't build the highest possible gradient. We build a gradient, but it's not the maximal gradient we can build, because we're not burning all the ATP we could possibly burn to make that gradient. So when we're highly hydrated, we build a gradient because we have the mechanism here. We've got the pump equilibration, flow equilibration going on, but we have less pumping, and so we have a lower gradient, not as much as we could have. Then the fluid, so because sodium and chloride is being pumped out of the ascending limb, what enters the distal tubule and what enters the collecting tubule is hypotonic. The salts, the sodium, has been extracted from it. So this is low osmolarity. And in the absence of vasopressin, which is another hormone that responds to our hydration state, so highly hydrated, low vasopressin. Where does vasopressin come from? The posterior pituitary. So our brain is detecting high hydration. It's detecting dilute blood, and it's saying, we don't need any vasopressin out there, and it's not secreting vasopressin. So low vasopressin means low water permeability here. Let's not worry about the fact that it's also transporting salt, but we're not transporting water here. That's the key thing. So a dilute input and no water permeability leads to a dilute output. You don't need to worry about the numbers. I'm not going to ask you what is the minimum osmolarity that we could produce, but that's about it. Okay, so in the hydrated state, low aldosterone builds a small gradient. Low vasopressin doesn't use the gradient, and the hypotonic fluid is excreted. Questions about what we're doing in the hydrated state? Okay, in the dehydrated state, low water in the body, we need to conserve it. So aldosterone is released. Aldosterone tells the ascending limb of the loop of Henle, make more pump. It increases the expression of the sodium potassium ATPase and especially the sodium 2-chloride cotransporter. And that change in expression is at the genetic level. It just increases the transcription of those genes, increased transcription of the genes, increased translation of the genes, increased protein expression. It takes some time, but we're talking about minutes, not days, maybe hours. Aldosterone increases the expression of the pump, so we're pumping more sodium out of the ascending limb. Okay?
Oh, I'm just going to make you really quickly with the two pumps, so that it's the sodium, potassium, ATPase, and
It's the same pumps as before. It's increasing the expression of the sodium potassium ATPase and the sodium potassium 2-chloride cotransporter. Sodium potassium ATPase is in the basolateral membrane, and the sodium potassium 2-chloride cotransporter is in the apical membrane. Together, they work to pump more of those pumps, more pumping. So now, isotonic input, lots of pumping and equilibrating and shifting. So pump, equilibrate, shift, equilibrate. That combination of things, repeated over and over again with a lot of pump, builds a very high gradient. with a very concentrated solute concentration in the interstitium deep in the medulla. High dehydration. Dehydration also causes the posterior pituitary to release vasopressin. Vasopressin is going to increase the expression of water channels. We're going to talk about those water channels in a few moments. Increasing the expression of water channels, more water permeability in the collecting tubule. So we have a collecting tubule that is going through this highly concentrated interstitium. It's highly water permeable. So if we have a dilute fluid moving, let's just say dilute-ish fluid, moving into a highly concentrated interstitium, and the membranes are permeable to water, water is going to flow. Now you could say, well, this flow of water is going to dilute this gradient. And in fact it will. But the point is, this machinery is working at full steam to maintain this gradient, even as this tubule is using that gradient to pull water out of the fluid. And so this water ends up going into the vasa recta to get recirculated back into the body. So the difference is aldosterone builds more pumps, sodium potassium ATPase, sodium chloride 2 cotransporter, more pumps, more of the counter current multiplier mechanism, builds a steeper gradient, a higher concentration here. Vasopressin tells the collecting tubule, make water channels, water permeability, water flows, a concentrated urine, minimizing water excretion, which is what we want to do if we're dehydrated. Questions?
Hearing this now, I feel like I'm deeply dilute here.
Okay. You want me to go back and tell the dilute story again?
Yeah.
In a dilute story, low expression of the pumps, low countercurrent multiplier mechanism, low building of the gradient, low vasopressin, low water permeability, so what gets excreted is dilute. Let me do it fast, we'll go back and forth. High dehydration, high aldosterone, high expression of the pump, high countercurrent multiplier. We still have a dilute fluid leaving this because we pumped all this sodium chloride out of it. This is still the same. But now this dilute fluid is moving through a water permeable tube, and it's moving into an interstitium that is highly soluted, highly concentrated with solute. So water is pulled from the fluid, and we get a concentrated urine. In this case, highly hydrated water excretion in order to reduce the overhydration. In this case of dehydration, highly concentrated urine in order to minimize water excretion.
So the osmolarity in the distal tubule is the same, but is there a volume difference because there's more solute being pumped out in the ascending?
You mean, why is this the same as this?
Well, I understand why it's the same because water moves with the solute.
Yeah.
But is there a lower volume when there's higher pumping?
Yes, there is lower volume. Yes, there is lower volume, and the reason I know that is one of the things that the macula densa detects, and we're going to get to that in a minute. The macula densa detects lower volume flowing in the distal tubule, and that's going to affect some changes. So yes, I'm certain that the volume flow in the distal tubule is lower in this setting than in this setting. Remember, the macula densa is at the beginning of the distal tubule, or the end of the ascending limb. So this, in a cartoon like this, you'd say, well, that's where the macula densa has to be. Other questions? That was a good one. That was helpful. All right, let's move on. We can come back to this during the breakout. If you have questions, in the small, I'm happy to try to clarify. The main point of understanding the vasa recta, is that the geometry of blood flow parallels the geometry of urine-ish flow, urinary fluid flow, and by paralleling it, the vasa recta preserves the gradient and reduces the washing out of the gradient. We take some solute. Remember, we are trying to reabsorb solute. We do take some solute. We want to take out some solute. That's part of reabsorption. But we don't want to wash out the gradient too much. And that's why the blood flow parallels the urinary fluid flow. That's really all. So you don't need to know the numbers here. It's just that the numbers are trying to illustrate that we have a highly concentrated solute and a dilute and a isotonic solute concentration in the blood, as well as in the interstitium. Interstitium, blood osmolarity, slightly hypertonic leaving, but not grossly hypertonic leaving. Because the grossly hypertonic leaving fluid would have washed out the gradient that we spent a lot of effort, both evolutionarily and ATP-wise, to build. Okay, so now I want to open up one of the black boxes. So what I mean by black boxes, you have been wondering, I think, maybe, how does aldosterone increase the expression of the sodium potassium 2 chloride and the sodium potassium pump on the ascending limb? You could well be wondering that, and that's a genetics question. It has to do with aldosterone activates transcription factors that increase the expression. So I'm not going to get into the genetics, the molecular biology. I'm just going to leave that as a black box. Aldosterone is a hormone that increases the expression by changing transcription factors in these cells that increases the expression of those pumps. That's enough for us. If you're a geneticist, that's not even close to enough. If you're a molecular biologist, it's like, what? But for now, for us, that's good enough. But I do want to open up the black box of how vasopressin increases the water permeability of the collecting tubule. Because that's physiology more than it is molecular biology. And here's the slide that illustrates it. So let's get oriented. Basolateral membrane. What else would be in this membrane if I wanted to illustrate another key protein in the basolateral membrane of all epithelial cells in the kidney? The sodium potassium pump has to be there. It's everywhere. This slide is illustrating something else, so it's not there, but it should be there, really. Here's the apical membrane. This is the tubular fluid in the collecting tubule. We're focusing on the collecting tubule. This is the interstitial fluid facing vasorectal capillaries in the medulla, in the collecting tubule. Okay? Here's vasopressin, the hormone. It was released from the posterior pituitary. It goes everywhere in the body, that's what hormones do. And these cells, the collecting tubule epithelial cells, express a receptor. It's a GPCR. It's coupled to GS, S as in Sarah. And now you're off and running with the GS story. I don't need to tell you about it, but there's GS alpha. What caused GS alpha to separate from the receptor? Well, binding, conformational change, GTP dissociation, GTP association, it's all the same story. Activated alpha activates adenylyl cyclase. Cyclic AMP, regulatory subunits, we've got some things missing here. Cyclic AMP binds to regulatory subunits, more catalytic activity, protein phosphorylation, right? This is PKA. PKA in this story phosphorylates two key things. It phosphorylates a water channel called aquaporin. It's got a helpful name. Aqua, water, porin, channel. Aquaporin is a water channel. It's a water-specific membrane protein that allows water to flow across the membrane. Aquaporin is expressed in these cells and is resident in vesicles that are sort of ready to be exocytosed and depositing the aquaporin into the apical membrane. So when aquaporin is phosphorylated, that's a signal to the cell saying, exocytose me. Now we have water channels. So water can move from urine into cytoplasm, and we have a constitutively active water channel over here. So in the absence of vasopressin, remember the absence of vasopressin means low water permeability. This channel is still there, but because there is constitutive endocytosis all the time, these channels have gotten all taken up into these vesicles, and there's very little aquaporin on the apical membrane. And if we have very little aquaporin on one membrane, then it doesn't matter how much aquaporin we have over here, we're not going to get transcellular water permeability. So, but in the presence of vasopressin, story, phosphorylation, exocytosis, high water permeability here, and high water permeability here means high transcellular water permeability. The other thing that PKA phosphorylates is a transcription factor. It's called CREB. You may have heard of this in cell-phys or in some other location. CREB is a transcription factor that is activated by PKA. And that leads to increased expression or synthesis of aquaporin. So we make more aquaporin, and then we exocytose more aquaporin in order to increase the expression of aquaporin in the apical membrane when there is vasopressin. And remember, now we're moving water from urine into interstitium and into blood.
Can you repeat what you said about
CREB is a transcription factor that is activated by PKA phosphorylation. And it increases, there are CREB response elements that increases the expression, the transcription and translation of aquaporin. So now that this black box, the vasopressin story, has been outlined in its molecular detail. Constitutive, vasopressin dependent. GS story again. Yeah.
How does the body like detect, like to release the vasopressin?
There are cells in the hypothalamus that are called osmoreceptors. They are sensitive to, they basically have stretch-activated channels. When those osmoreceptors are in a dilute fluid, they stretch a little bit, and that shuts off the secretion of vasopressin from the cells. And if those cells are in a hypertonic solution, they shrink a little bit, and that tells the cells, I'd have to review the mechanisms, but that cell tells the osmoreceptors in the hypothalamus to release vasopressin from the posterior pituitary. So there are specific hypothalamic osmoreceptors that trigger the release of vasopressin from the posterior pituitary. So we've talked about baroreceptors that are sensitive to pressure, chemoreceptors that are sensitive to oxygen or CO2 or acid, and now we're talking about osmoreceptors. I will not test you about osmoreceptors because I'm leaving that as a black box. When I say black box, do you know what I mean when I say black box? It happens, but we're not going to open it up and try to explain what goes on inside. I need to mention urea, because if I don't mention urea, the professors that taught me renal physiology will be tearing their hair out. And like me, they don't have very much of it. So I owe it to them to tell you about urea. But I don't want to dwell on it because it's complicated. But you know that urea is in urine, so let's just talk about it. This is the slide that reminds me to talk about it. But all I really want you to know is that urea is a solute that is produced by the metabolism of protein. So protein, if you eat protein, you make urea. And we all eat some protein. So urea is a solute that comes from metabolism of protein in our diet. Urea is transported in just about all of the segments of the kidney. So it's transported, it is freely filtered. So it gets into the urinary fluid through filtration just like other small molecules. It is transported in all of the segments of the nephron: proximal tubule, descending, ascending, distal, collecting tubule. It's transported in all of those to varying extent by passive transporters, urea uniporters. Remember we talked about uniporters in cell-phys. These are passive transporters of solutes. They're not channels like aquaporin or like sodium channels. They're transporters. They bind a solute, conformational change, bind a solute on one side, conformational change, release the solute on the other side. Remember that story of binding, conformational change, unbinding. Those are the urea transporters. What I want you to know is that urea, so some of the urea that gets into the collecting tubule is taken out of it by passive transport, and it contributes another solute. This is the key thing. Urea contributes another solute to the medullary solute gradient. So I've been pretending all this time that it's sodium and chloride, because this slides like this is focusing on sodium and chloride as solutes that build up this gradient. But here is little urea. So this, I could have done it right here, but I kept it separate on this slide. Urea contributes to the solute gradient. But it's all through passive movement. So we retain some urea in order to contribute to this gradient so that we can conserve water when we need to. But we always need to excrete some of it because we need to get rid of the metabolic waste product from protein metabolism. So we have a finely tuned system for retaining some of it to be used as a solute and excreting some of it because we have to get excreted as a waste product. So it's pretty interesting. We use a waste product as a solute to help us conserve water. But we have to get rid of some of it because it's a waste product. That's all I really want to say about urea. If you start to ask me about what are all these numbers here, we will all get confused. So don't ask me. Sophie.
If you have like, can't you eat like too much protein and then like it be toxic? Is like
Yeah.
urea a component of that, or?
So you're talking about gout. Eating a very rich protein diet causes uric acid, which is another byproduct of protein metabolism, to accumulate in joints. And so it's not urea, it's uric acid that builds up in joints and contributes to the disease that we call gout, which is pretty rare these days, but was once upon a time pretty common.
Is urea like just a different byproduct than uric acid? Can urea become uric acid, or?
I don't know whether urea can be metabolized to uric acid or vice versa. I don't know if those are different pathways from protein or whether they are part of the same pathway or interacting pathways from protein. I'd have to look that up. Other question? Yes?
Do you know, like, in regards to like evolution, why we made it so that we reabsorb it and use it if it is a waste product? Like, why
Yeah, so-
would we do that?
I mean, so I'm such a poor evolutionary biologist that I can't, those kinds of questions almost always stump me. I know why we make urea, because the actual product of protein metabolism is ammonia, and ammonia is toxic. So we convert ammonia into something less toxic, urea. We actually spend a lot of ATP doing that. But why we evolved a system for using the waste product to help us, I don't know the answer to that. But you can see why it would be helpful to have another solute. So in addition to having 1,200 milliosmoles of sodium chloride, if we added another 500 milliosmoles of urea, now we can make an even more concentrated urea. And as long as we excrete some of it for some of the time, we're okay. So let's talk about these control mechanisms, right? So this is sort of a summary slide. The loop of Henle builds and the vasorecta maintains the solute gradient. That's a key concept. Variable water permeability allows us to either make a concentrated urine or a dilute urine. In the presence of vasopressin, high permeability in the medullary collecting tubule, high water absorption, concentrated urine, low vasopressin, low water permeability, dilute urine. We're gonna use that in a moment. Oh, this is the slide because why does urine have color? It's just sort of an interesting aside. This will not be on any test, but now you can tell your 7-year-old nephews why their urine is yellow. is yellow, right, but it's concentrated enough. None of these elements that's in it is pigment. It's all clear. Urea is not the reason why urine is yellow. Urea is colorless as well. What gives urine its color is heme. The breakdown of heme, you can see that these ring structures broken out and linearized is a yellow color. So heme is a red color, and broken down heme is a yellow color. And in fact, I'm going to say it now because I'm not going to say it when we get to the digestive tract. But the digestive tract also, feces have a color, it's brown, and that's another pigment from heme breakdown. Okay? So that's why urine is yellow. It's got nothing to do with urea. So getting back to control, now we're back on track. Remember that here, this sort of looks like one of those worms from Dune, right? But this is the distal tubule, macula densa, communicating with the afferent arteriole and juxtaglomerular cells. Remember that fluid in the macula densa, fluid in the distal tubule, tells the macula densa to tell the juxtaglomerular cells to release renin when this fluid flow is low. So that was getting back to Sarah's question a few minutes ago. Low flow, macula densa, juxtaglomerular cells, renin, angiotensin, aldosterone. Now you know where the aldosterone signal came from. We're still leaving in the black box how aldosterone works, but that's how aldosterone was turned on. Do that again. Low flow, macula densa, juxtaglomerular cells release renin. At the same time, the sympathetic nervous system is constricting the afferent arteriole. So I should have said this at the upfront. When I talk about low flow and constricting of the afferent arteriole, what are we talking about? A highly hydrated situation, or are we talking about a highly dehydrated situation? Dehydrated. Dehydrated, we're working hard to conserve water, constricting the afferent arteriole to reduce filtration. Reduced filtration produces low flow. Low flow then goes to produce aldosterone by several steps, which helps us conserve water by building that solute gradient. So I'm trying to put it all together. Sympathetic nerves, constricting afferent arterioles. What G protein story am I thinking of when I think of afferent arterioles, when I think of sympathetic neurons constricting afferent arterioles? GQ. What neurotransmitter? Norepinephrine and epinephrine. GQ story, contraction of the smooth muscles, constriction of the afferent arterioles. So here's just the word version of it. Macula densa stimulates the juxtaglomerular cells to release renin. Did I give you the slide as a handout? So you can cross out prostaglandins. I'm not going to ask you to know that prostaglandins are part of the story. Just trying to simplify things because we're going so fast. Macula densa stimulates the juxtaglomerular cells to release renin. Renin is going to do stuff on the next slide. At the same time, the sympathetic nervous system constricts the afferent arterioles And I guess, and it also stimulates renin release. In the dehydrated, we're still in the dehydrated setting. So here is a slide from endocrinology. I don't want you to know the details, but I want to open up this black box a little bit. So here's renin. It catalyzes a reaction from a precursor. I want to introduce this language, angiotensinogen. If you see the word ogen attached to anything else, it tells you it's a precursor protein. But we're going to see that in the digestive tract big time. So angiotensinogen is a precursor, and renin cuts it into a product. This product is converted by ACE, A-C-E, angiotensin converting enzyme. ACE converts angiotensin into the active hormone, angiotensin 2, which activates aldosterone. I mean, it tells the adrenal cortex to make aldosterone, which then has its effect on the ascending limb of the loop of Henle, just that I've been telling you about since the beginning of this hour. We're not going to talk about angiotensin's effect on the cardiovascular system. It's for another day, or maybe another year. Okay, questions about this pathway from juxtaglomerular cells to a hormone, angiotensin, to another hormone, aldosterone, to water conservation. Questions about that?
I was just gonna say, like we talked about this in analogy with COVID, how like the ACE system could.
Yeah, so COVID, so I haven't been reading up, but COVID binds to the ACE receptor.
Yeah.
So it binds to, it gets inside our lungs by binding to a protein whose physiology is to bind ACE, is to localize ACE, angiotensin converting enzyme, into the endothelium. So this is an enzyme that is secreted from endothelial cells and then stuck there so that it's working on the blood. But there's always endocytosis and exocytosis going on, and the virus gets into our cells by hijacking the endocytosis story of the ACE receptor. Yeah, so we're now talking about COVID outside of my league. Okay, let's continue talking about control. So the remaining few slides are sort of flow charts that I think are helpful in review. So let's take this story. Blood volume. How could you have low blood volume? So that's the story. Low blood volume. How could you, under what physiological scenarios or pathophysiological scenarios can you imagine low blood volume?
Hemorrhage?
Hemorrhage? Bleeding?
Just the blood.
Okay, you've given blood, but that's essentially the same as bleeding. What else? One more thing that we've been talking about for an hour that would lead to low blood volume.
Dehydration?
Dehydration. So low blood volume could come from dehydration or from loss of blood. So let's start with the sympathetic nervous system because it's kind of simple. Sympathetic nervous system is low blood volume is detected by the hypothalamus, which tells the sympathetic nervous system to release its hormones, norepinephrine and epinephrine. They trigger the release of renin. Now here's the renin story. Renin, angiotensin, aldosterone. So aldosterone increases the reabsorption of sodium at the loop of Henle and assists in the reabsorption of water. Reabsorption of water is going to decrease excretion or retain water, which will increase blood volume and try to correct the defect, whether it's intentional by donating blood or non-intentional through an injury or dehydration. And we can do other stories. Low blood volume, low blood flow, low GFR, low macula densa activity, and then the same story. Let's not get into the arterial pressure story because there are stretch activated parts of the story. The point is, so the kidneys are evolved to control volume as a way of helping us control water levels, salt levels, and blood pressure. So you're going to, in the problem set, you're going to work on some diuretics that are used for the control of blood pressure. So this is the key concept here, is that controlling blood volume can help control blood pressure, especially in hypertension, chronic high blood pressure. So everything that's going on here can be flipped upside down. If we have high volume, high pressure, decreased renin, decreased aldosterone, decreased pumping, more excretion of water. We have mechanisms for doing that, and we have drugs that help us do that. So you're going to be working in the problem set on diuretics. Diuretics are drugs that are designed by humans to increase diuresis, which means increasing the production of urine in order to reduce volume, in order to reduce pressure. Okay, so that's leading you into what we're going to be doing pretty soon, which is going out to our breakouts and working on the problem set. Volume. So again, this illustrates volume can go up or down. Pressure is related to volume. So high volume is associated with high pressure, low volume is associated with low pressure. Pressure and osmolarity lead to vasopressin release from the hypothalamus, from the posterior pituitary. Vasopressin, permeability, water excretion. We can do it this way, so low plasma volume, high osmolarity, say, or just low volume, triggers vasopressin secretion. Water reabsorption is maximized. Water excretion is minimized. But we can do it the other way around. High plasma volume, high pressure, low vasopressin secretion, high water excretion in order to reduce too high blood pressure. So these stories, maybe the key point here is that these stories illustrate one story, low volume, but you can flip all the arrows the other way around to describe another story. This is a complicated slide that puts the cardiovascular system